students, dear guests, and welcome back again on the YouTube channel of University of Luxembourg. It is a big pleasure today for me to introduce our guest speaker, who is Dr. Schockmel, specialist for infectious diseases with a huge experience in research in microbiology. And for those who are intending to do their bachelor's degree in medicine at our university, Dr. Schockmel will be one of your teachers for infectious diseases and microbiology. We apologize for the technical problems we met last week, and which made us defer by one whole week, but this allowed us to check up the whole system, and hopefully, let's cross fingers, it will work today. The topic is, of course, crucial. The topic is dealing with vaccination against COVID disease, which is so far an expectation, a wish, because it will be the ultimate possibility to win the battle, which we, let us say, only we were only able to freeze it with the confinement, but of course we all know that the virus is still around, that we should remain very careful. Gérard will give us his talk, which, as I know him, is expectedly most thoughtful. And after his talk, we will have a surprise for you. We will have two guest discussants whom you know well. You certainly remember Professor Wong from Fudan University, who gave a brilliant talk on epidemiology of COVID some 2 weeks ago. And he will be joined by Professor Liu, who is also a specialist for epidemiology and who was the discussant for Professor Wong's talk. So I invite now Gérard to take over, and I get back to you to launch the discussion in some 45 to 60 minutes. I hope you will enjoy.
Well, thank you, Gilbert, for the introduction. Hello to everybody and welcome. So I suggest that we go straight into our subject of today, which is about coronavirus vaccines. With more than 5 million cases of coronavirus infection reported worldwide and over 300,000 deaths, the pandemic caused by the novel coronavirus is unprecedented. The humanitarian crisis and the economic crisis already affect more than a third of the world's population. Hundreds of millions of people risk to lose their jobs and to be unemployed. In the face of such a threat, to have an effective vaccine against the coronavirus has become a matter of urgency. So, what would you expect from a coronavirus vaccine? A vaccine stimulates the immune system so that the vaccinated person develops a protective immunity against the virus. This immunity can either prevent infection or prevent severe symptoms in case of infection. How does the vaccine stimulate the immune system? By presenting viral proteins to the immune system. These viral proteins can be presented either on the surface of a particle, that is, for example, the surface of a non-pathogenic virus, the surface of a virus-like particle, the surface of a human cell. Viral proteins can also be presented either in an encapsulated form or bound to a carrier structure. A vaccine needs to protect the individual and the community. Ideally, you would want to have a vaccine that does not need a cold chain, because if there is a need for refrigeration, this can be problematic in Africa and in developing countries. Ideally, you would want a vaccine able to achieve full protection with a single dose. Because if you need 2 or 3 doses, as in a classical vaccination scheme, then the degree of protection achieved with the first dose can only be partial, and you are going to lose precious time to vaccinate everybody a second time and a third time. You would want a vaccine that remains effective in case the coronavirus mutates, and that provides some cross-protection against other coronaviruses, thinking of SARS coronavirus, MERS coronavirus, or emerging coronavirus. The vaccine also must be able to be administered to persons at risk. Persons at risk, like persons aged above 60 years, diabetics, persons with hypertension, immunosuppression, healthcare professionals, children. You would want to have a vaccine that you can use for mass vaccination programs. You would want a vaccine that is accessible to the most vulnerable, because they are the hardest hit by the pandemic. And here I'm thinking of homeless people and also of people who don't have health insurance. You might know that 10% of Americans don't have health insurance. And very often health insurance for them comes with their jobs. So if they lose their job, they will not be able to afford health insurance anymore in many cases. Finally, you would want the vaccine to be available to populations in resource-poor countries. Altogether, it will probably be necessary to develop a variety of vaccines based on different vaccine technologies to satisfy the individual needs of vaccine recipients. Individual needs in terms of risk profile, individual needs in terms also of age groups. There's a difference between someone aged 90 and a newborn baby. Also, you need to satisfy a global demand for a coronavirus vaccine. And that is why I think that you probably need a variety of vaccines to satisfy this demand. What kind of immunity do you want a vaccine to achieve? We said that the immune system is stimulated by viral proteins, particularly by the spike protein or by fragments of the spike protein. Ideally, the regions of the spike protein that generate an immune response are conserved, that is, not prone to mutation or to genetic recombination. An optimal protection against the virus requires two things. You need antibodies and you need immune cells to protect you. I mean, most of us, when we think of vaccination, we think of antibodies. So that because of the vaccines, we develop antibodies, and these antibodies will protect us. And it is true that indeed, antibodies that are rapidly produced thanks to prior vaccination attack the virus as soon as it enters the human body. However, once an infection has been established, the action of antibodies might be insufficient. The virus can pass from one cell to the other cell via intercellular contact, not exposing itself to the extracellular environment that contains the antibody. So once infection has been established, you need immune cells to control and to clear infection. Immune cells that destroy infected cells to stop the replication of the virus and to eliminate the virus. Now, in the laboratory, it is much easier to determine the presence of antibodies in blood than it is to determine activity of specific immune cells against the virus. Now, if you want even more antibodies or you want even more immune cells, what you can do is to add an adjuvant to your vaccine. Classical adjuvants are, for example, aluminum salts. Adjuvants allow you to even boost the stimulation of the immune system with your vaccine. So with adjuvants, you can reduce the dose of the vaccine. In this way, out of the same quantity of vaccine substance, you can produce a higher number of vaccine doses, and thus you can vaccinate a higher number of people. So adjuvants clearly are very attractive in many circumstances. Let's now talk about the different type of vaccine technologies. You have received a graphical guide, and I think it is quite well done. So let's start with classical vaccines. Classical vaccines use the coronavirus itself. And there are two types of classical vaccines. There are the live attenuated viruses, and then there are the inactivated viruses. So let's talk about the live attenuated viruses first. They use weakened coronavirus, but they remain capable of replicating in host cells. The replication of the virus generates a strong immune response, similar to the immune response generated by wild-type coronavirus. So a single vaccine dose may be sufficient. However, development of these types of live attenuated virus vaccines is slow because you need to do extensive safety testing to prove that these vaccines are safe for the vaccine recipient. Live attenuated virus vaccines are contraindicated in persons who are immunosuppressed. In terms of safety aspects, what you need to make sure is that live attenuated viruses don't undergo mutation or exchange of genetic material with other live viruses. This has rarely occurred with live attenuated poliovirus vaccines, and it is always a theoretical risk to be considered, even if it is probably quite rare. So there is a second type of classical vaccines, and it is inactivated virus vaccines. Inactivated virus vaccines are non-infectious and don't replicate. Viral proteins expressed on the surface of the inactivated virus generate an immune response. Now, inactivated virus vaccines are less powerful than live attenuated virus vaccines because there is no production of viral proteins in the body. The totality of viral proteins is administered with the injection. This explains why generally with inactivated virus vaccines, you need several vaccine doses to achieve protection. Also, when you want to produce inactivated virus vaccine, you need large quantities of virus to start with. The advantage of inactivated virus vaccines is that they can be safely administered to immunosuppressed patients. There are at least 7 teams working on these classical vaccines. Now we come to a different kind of vaccines, vaccines that are produced by genetic engineering. Now, genetic engineering is great for researchers because it allows them to actively design their vaccines. So if they want a viral protein to be produced by a cell, well, they will just put in a gene that encodes for that protein. If they are confronted with a viral gene that confers virulence, well, they just take it out and there's no more virulence. So the big advantage of genetically engineered vaccines is that you can actively design them. The first of such vaccines I want to mention is the whole virus vaccines, viral vector vaccines. So what are viral vectors? I mean, a viral vector is a non-pathogenic virus, for example, adenovirus, that has been genetically engineered to instruct cells to produce coronavirus spike protein, for example. When the spike protein gets exported from the cell, it will elicit an immune response, and generally you don't need adjuvants with this kind of vaccines, and a single vaccine dose may be sufficient. There are two types of viral vector vaccines. There are the non-replicating and the replicating viral vector vaccines. Non-replicating viral vector vaccines are such that the viral vector is incapable of replicating. Most coronavirus vaccines in development that use viral vectors are actually using non-replicating viral vectors. And then there are the replicating viral vector vaccines. Here, the viral vector is a weakened virus, but that remains capable of replicating in host cells, and thus generates a strong immune response. Viral vector vaccines are very attractive because they can be designed to instruct the cell to build the protein of any infectious agent, be it Ebola, be it coronavirus, be it flu virus, be it salmonella, whatever. In terms of safety aspects, viral vector vaccines are GMOs, which means they are genetically modified organisms, and as such, they are subject to strict regulation. Also, right now, we don't know in how far these vaccines will affect the metabolism of cells that produce viral proteins, or in how far they will affect the processing of immunogenic fragments of viral proteins by specialized immune cells. There is also a slight risk that such vaccines might integrate into the host genome, and this has another related risk, potential risk, that would be oncogenesis. I think this risk is probably extremely small with non-replicating viral vector vaccines, but with replicating viral vector vaccines, you really must take care to design them in a way such that this risk is clearly eliminated as far as possible. Viral vector vaccines can be less effective if the vaccine recipient already has a pre-existing immunity against the viral vector. That is one of the reasons why the Oxford viral vector vaccine uses an adenovirus from the chimpanzee, because human immune systems are generally unfamiliar with chimpanzee adenovirus. There are at least 25 teams working on viral vector vaccines. A second type of genetically engineered vaccines are protein subunits vaccines. You can use either whole spike protein or fragments of whole spike protein, or even very tiny fragments that you would call peptides of spike protein. The advantage with peptides is they are so small that you can actually produce them by synthesis, whereas protein and larger fragments of the viral protein needs to be produced by cell culture, which is much more work. These viral proteins, typically spike protein, are very often presented in a trimeric form. That means there are always 3 of them, just as for the coronavirus. So you have 3 identical spike proteins sticking together, bound to some common structure, or microencapsulated, bound to a carrier, as I said, or with a slow release formulation. Protein subunit vaccines are non-infectious and don't contain any genetic material. There are many of them on the market already, and you might actually already have received one or the other of them, such as vaccines against hepatitis B, against varicella, against influenza. As these vaccines are composed solely of proteins or protein fragments, they often are unable to generate a sufficiently powerful immune response to confer complete protection against the virus. That's the reason why with these vaccines, you generally need to administer several doses, and you also need to add adjuvants. The primary objective pursued with these vaccines is to create sufficient protection for people at risk of infection, such as healthcare workers, while waiting that more powerful vaccines become available. There are at least 28 teams working on protein subunit vaccines. So now now we are going to talk about the third type of vaccines, so-called virus-like particles. Virus-like particles are particles that are non-infectious and consist of empty shells devoid of genetic material. Viral-like particles carry on their surface coronavirus spike proteins to generate an immune response. As these particles imitate infectious virus particles, they are able to generate a strong immune response in the absence of adjuvants. A single vaccine dose may be sufficient. Virus-like particles are more complex to produce than protein subunit vaccines. There are at least 5 teams working on virus-like particle vaccines. A novel vaccine technology that has received a lot of media coverage and attention are the so-called nucleic acid vaccines. Nucleic acids, as you know, is messenger RNA or DNA. Nucleic acid vaccines instruct the cells to produce coronavirus proteins, typically the spike protein. When these proteins get exported from the cell, they generate an immune response. Nucleic acid vaccines hold great promise because these vaccines are simple to design. Basically, if you know your protein, you know the genes encoding for the protein, you can design such a vaccine in a single day. They are also simple to produce because they are produced by synthesis. You don't need cell culture, and thus they can be made rapidly available. Also, like viral vector vaccines, nucleic acid vaccines can instruct the cells to build the proteins of any infectious agent, be it Ebola, flu virus, coronavirus, salmonella, whatever. However, at the current time, there is not a single licensed messenger RNA or DNA vaccine yet, and we don't know for the time being whether they will be safe and effective. So there are 2. Let's talk first about messenger RNA vaccines. In the cell, you may remember from biology, messenger RNA is a messenger between the cell nucleus and the cell cytoplasm, in charge of translating genetic information into proteins. Used as a vaccine, messenger RNA instructs the cells to produce coronavirus spike proteins, for example. When these spike proteins get exported from the cell, they will generate an immune response. Messenger RNA vaccines have to be administered directly into the cells. For that, you use formulations either on the basis of liposomes or of lipid nanoparticles. Lipid nanoparticles, in particular, not only facilitate entry of messenger RNA into the cell, but they also have an adjuvant effect. So that means they reinforce immune stimulation caused by the messenger RNA. Safety aspects. Well, first, there is the inherent instability of the single-stranded messenger RNA. So here you need to develop the technology to make these molecules stable. Also, you need intracellular administration that, again, requires development of a specific technologies. Now, these technologies have not been used in humans, and so that is another unknown that needs to be tested. You don't know also how these vaccines will affect the metabolism of cells that produce viral spike proteins, or how they will affect the processing of immunogenic fragments of the spike proteins by specialized immune cells. The second type of nucleic acid vaccines are DNA vaccines. Now, DNA vaccines need to be administered into the cell nucleus. The DNA vaccine contains the instructions for the synthesis of coronavirus spike proteins. These instructions are transcribed into messenger RNA, and the translation of messenger RNA induces the synthesis of coronavirus spike proteins. Exported from the cell, these spike proteins will generate an immune response. In contrast to messenger RNA, DNA, which is double-stranded, is a very stable molecule and able to generate a strong immune response. An important challenge with DNA vaccines, however, is the fact that they need intranuclear administration. For that, you use a technique called Electroporation. Here you apply an electric field onto the cell membrane, and this makes the cell membrane become permeable, allowing entry of the vaccine DNA into the cell and then hopefully into the nucleus. Safety aspects. There is a risk, a specific risk, with DNA vaccines that the vaccine DNA might integrate into the host cell DNA. And that always comes with a second risk, which would be a risk of oncogenesis. Again, you don't know how cells that will produce viral proteins see their metabolism affected by these vaccines, and how immunogenic fragments of these viral proteins are processed by specialized immune cells. And last type of technology, which is not mentioned in your graphical guide, are vaccines that use human cells, particularly the human cell line K562. These human cells have been irradiated, so they are non-replicating, but they have been genetically engineered to express on their surface coronavirus spike proteins. I think this comes to no surprise to you. Now, these vaccines also generate strong immune responses, and a single vaccine dose can be sufficient. There is already experience in humans with the cell line K562 expressing a human protein due to genetic engineering on their surface, so there are already safety data available for this kind of vaccine. Right now, when I was presenting the different vaccine technologies, I also said a few words about safety aspects. However, there's more to be said about safety effects, and that is what I shall do next. So, one important safety aspect to be considered in all vaccine developments is the potential for disease enhancement. Disease enhancement means that vaccinated people get a more severe disease than people who have not been vaccinated. So obviously, this is exactly the opposite of what you want. You want a vaccine to prevent you from developing severe disease. You don't want a vaccine that causes severe disease rather than the opposite. So what can be done to avoid, to recognize early on whether your vaccine could have disease enhancement effects? I mean, there are 4 things that need to be checked for. The first one is you need to make sure the type of immune response generated by your vaccine. You can do that by checking on the molecular signals that will be secreted and circulated in blood when you give your vaccine. And depending on the pattern of these molecular signals, you know whether you have got the right immune response or not. A second point is obviously that you will check that there is absence of tissue damage caused by the vaccine you are administering. The third factor is that you must make sure that the viral proteins, particularly the spike proteins, are correctly folded in space. Because if they are not correctly folded, you are not sure of the type of immune response that they will generate. And the fourth factor is that you want to make sure that your vaccine elicits sufficient numbers of neutralizing antibodies. Neutralizing antibodies are antibodies that prevent viral replication. So with these four factors in mind, you can actually be pretty sure either to early detect that there's a problem, that you have disease enhancement with your vaccine, or that there's no problem and you can proceed safely into your development. There is another risk. It is more of a theoretical risk, but unfortunately, people who don't like vaccines very often allude to it. So I have to mention it as well. It is the risk that the vaccine might cause an autoimmune disease by mistaking the virus or taking own body tissue for the virus. This is not an effect we have reason to believe is important with the coronavirus, neither with the natural coronavirus infection, nor with vaccine against coronavirus, but still it is something obviously you have to watch out for. Let's talk a little bit about animal studies. I mean, you know how it works with animal studies. The animal receives the test vaccine, and after that, the animal is challenged with live coronavirus, so infected. After a while, then, animals are euthanized to study the effects of infection in their organs. Right now, it is not quite clear which animal model is best to study coronavirus vaccines. There are animals such as hamsters or ferrets that naturally get coronavirus infection. You can also produce mice transgenic for the human ACE2 receptor. And then there are rhesus monkeys. You might think that rhesus monkeys are very close to humans, which generally is certainly correct. However, rhesus monkeys naturally only develop mild symptoms of coronavirus infection. So if you are already in the phase of very mild symptoms in the absence of a vaccine, it is difficult to prove that your vaccine makes a difference. So the recommendation here is that you certainly should test your vaccine candidate in different animal models. And also, there are different strains of virus, and it is certainly useful to study the different strains of viruses, because there may be differences in their characteristics. If we proceed now to human studies, I mean, there is phase 1. This is the first study that you would carry out in healthy volunteers. Here, it will be important that before you administer the test vaccine to a healthy volunteer, you make sure the person to be vaccinated does not already carry anti-coronavirus antibodies in his blood. You need to know that, because if there are already pre-existing anti-coronavirus in the future vaccine recipients, then that gives way to a different evaluation of the effect of your vaccine. And also, it would allow you to detect potential disease enhancement if that mechanism turned out to be a problem. If you look at the large phase 3 studies that generally involve thousands of participants, here it is important that you have a sufficient number of participants in these studies, because in conventional phase 3 in vaccine development only detects adverse effects that are relatively frequent, meaning that are more frequent than one such adverse effect per 1,000 vaccine doses. So this means that an adverse effect that is more rare but potentially severe can just not be detected. Or if you would detect such an adverse effect, you would just consider that it is non-statistically significant, which is always a problem with phase 3 studies, because their statistical significance and the number of participants is generally calculated with respect to the primary efficacy parameter and not to study specific adverse events. It would also be prudent to have a prolonged follow-up of the participants in clinical studies. It would be useful to study re-exposition. By that I mean a person that has received a coronavirus vaccine might after that become infected with the coronavirus once, twice, several times, and you want to make sure that things happen as planned in that case. Also, it is possible that you are going to give a coronavirus vaccine to a person who already has been exposed to the coronavirus. Here again, you want to make sure that there are no problems. Another thing to make sure is that there is no interaction between your coronavirus vaccine and other vaccines that the person might receive, particularly elderly people might receive a certain number of vaccines. Children, anyway, receive a large number of vaccines. You also would want participants in clinical studies not only to be young and healthy, because if you only include young and healthy people in clinical studies, they may not be exactly very representative of the general population because they haven't got risk factors, and anyway, most of them will have a mild disease even in the absence of a vaccine. Okay, I will carry on. Let's now talk about global vaccine development. Here there are two large countries ahead of the lot. I'm talking about the United States and I'm talking about China. Now, if you look at the regions of the world, you have like 50% of vaccine developments taking place in North America. The other 50% are relatively equally split between China, Asia without China, you have Japan, South Korea, India, and Europe. You have big pharmaceutical companies and you have small startups. You have now about at least 90 vaccine candidates in development, of which at least 8 have already been administered to healthy volunteers. This does not mean that phase 1 studies in healthy volunteers are finished, but it means that they have already been administered to healthy volunteers. The other vaccine candidates are still in the preclinical phase. Timelines. The question you wanted me to ask right at the beginning when we discussed treatments for coronavirus was, well, will we have a vaccine in time? With more than 200 countries worldwide affected by the pandemic, this remains uncertain. Confinement and other sanitary measures, such as social distancing, hand hygiene, or face masks, certainly slow down the spread of the pandemic. But the pandemic will continue as long as the herd immunity remains below 70%. Now, where are we right now? There are now studies that have come out from Spain and from France, and the estimation is that right now the herd immunity in these two countries is about 5%. So we are very far away from the 70% required really to slow down the spread of the epidemic. Developing a vaccine traditionally has taken far over 10 years. For Ebola, the first virus that has been developed, the first vaccine, was developed in a record time, and the record time that was within 5 years. Now for COVID-19, what you would want and what certain vaccine developers aim at is a development of 1 year, perhaps even less than 1 year. That's highly ambitious. It's not totally unrealistic, but I'm going to talk more about that. There are indeed good reasons to be optimistic in terms of an accelerated vaccine development. And I give you the good reasons for that. I mean, one of the good reasons are the new vaccine technologies. We already discussed about that. We mentioned that viral vector vaccines, and specifically also messenger RNA vaccines, can be very rapidly designed, produced, certainly for messenger RNA vaccines, and made available. Second, there is also the fact that the novel coronavirus benefits from the experience with the SARS coronavirus. Third, there's the fact that drug agencies are prepared to consider accelerated development programs. Fourth, you have a parallel development of multiple vaccine candidates. Fifth, there is an unprecedented international coordination for vaccine development. And six, the financial resources made available are just extraordinary. So we talked about vaccine technologies, and now I'm going to talk about these factors that give way to some realistic optimism as regards fast vaccine development. I said that the novel coronavirus benefits from the experience with the SARS coronavirus. Yes, because indeed, the SARS coronavirus and the novel coronavirus are almost to 80% identical. They have very similar spike proteins, and they bind to the same cellular receptor. The vaccine candidates that were developed against the SARS coronavirus included live attenuated virus vaccines, inactivated virus vaccines, and also protein subunit vaccines. These were tested in animals, and so there is already considerable experience in animals as regards these vaccines. Unfortunately, they did not proceed to a stage where they were tested in humans, because then SARS disappeared and the money dried out. Also, the novel coronavirus benefits from experience with Ebola viral vaccines and also vaccines at an early stage developed against MERS coronavirus. A third factor for accelerating, speeding up vaccine development is the fact that drug agencies are prepared to consider accelerated development programs. I mean, you might know that for clinical development of the vaccine, generally there are three phases. You have phase 1 to start with. Normally, these are small studies with dozens of participants in each study. Then you have phase 2. They are larger studies where you would have hundreds of participants in each study. And finally, you have the large phase 3 studies, where in the studies you would expect not millions, but thousands of participants. Now, there are some good, and perhaps you might tell me some less good ideas how to speed up clinical development. I mean, one thing you could do would be to reduce the number of participants in clinical studies, either in the active arm, those that receive the vaccine, or in the control arm, those that receive placebo. Another way of speeding development would be to use simplified development programs. For example, not having a placebo control. Instead of a placebo control, you could just use data out of the scientific literature. Then there is the idea that you could have volunteers that would agree that they be vaccinated first and then challenged with live coronavirus. In this way, you would obtain very rapidly information on the safety and the efficacy of your vaccine. Another way of speeding up development would be to have overlapping phases. For example, with the Moderna messenger RNA vaccines, they are in phase 1, healthy volunteers, and at the same time still carrying out animal studies. The same thing with the DNA vaccine by Inovio. They are also in phase 1, healthy volunteers, but still carrying out animal studies. So here you would have an overlap between preclinical phase and phase 1. Another possibility is to combine phases, particularly to combine phase 2 and phase 3, as is planned for the Oxford vaccine. Now, the drug agency can authorize an accelerated development plan and can also commit itself to fast-track review and approval of the vaccine. The drug agency could even grant an emergency use authorization, allowing you to use the test vaccine from phase 2 in patients who are at very high risk of getting infected with the virus. Then the data collected in these persons would constitute phase 3 information. This has never happened so far, but it is certainly something that is being discussed right now. To make sure drug agencies would not accept vaccines with incomplete safety and efficacy data that would expose participants in clinical studies and later on vaccine recipients to risks that are potentially avoidable. That is why it is in the best intention of vaccine developers to hold prior consultation with drug agencies discussing the development plan they would like to propose for their vaccine candidate. Number 4 reason why you can expect a fast vaccine development is that in parallel, there are now multiple vaccine candidates being developed. Obviously, if you develop in parallel 10 to 20 vaccine candidates, you increase the chance that at least one of those vaccine candidates will be licensed in the end. The Bill and Melinda Gates Foundation has declared that they will finance the parallel development of 7 vaccine candidates. I think that's quite impressive. Another reason for expedited vaccine developments is international coordination. This coordination is actually unprecedented, but it is necessary because the challenges are enormous. What are the challenges? I mean, if you want to test your vaccine, you need to be there where the outbreak occurs. If you had wanted to test your vaccine in China, Wuhan, 3 months ago would have been a perfect timing. If you would want to have it tested in Italy, 2 months ago would have been perfect. But we don't know right now where the next outbreak is going to occur. And when there is an outbreak, there will be fierce competition because many manufacturers want to test their drugs to see whether they can cure or at least treat the COVID disease. Vaccine developers want to test their vaccines, and so there's competition between everybody. Then there's competition for material resources. Your vaccine probably will come in a vial, and each vial will need a rubber stopper. So already now we need billions of vials and we need billions of rubber stoppers, and the industries producing those are at their limits already before it even has started. You need important financial resources. I mean, normally you would expect hundreds of thousands US dollars to be required to develop a single vaccine candidate. Then what you need is access to technologies and to information, because technologies can come with a patent. And if you want to have access to the patent, you need a license agreement and you need to pay license fees. However, some companies don't file a patent because they just don't want to reveal information that they want to keep secret. In this case, this information is just not accessible to you. There's competition for manufacturing platforms. And obviously another challenge will be the comparison of data from very different studies. So what is needed is coordination. Coordination, you have the WHO, the World Health Organization. They have designed what they call a solidarity vaccine trial using a master protocol, an adaptive design, prioritization of vaccine candidates, and a single placebo group. Now that's very intelligent, because imagine you have independent vaccine developers developing 10 vaccines, having 10 studies running. Now, if for each study you have a placebo control group, you end up with 10 placebo control groups. However, with a single vaccine control group, you would have most of your patients included into the active arms of the studies where people receive a test vaccine. So having a single placebo control group is highly efficient in terms of recruitment of participants. That is for the WHO. Then you have the National Institutes of Health in the USA, and they have a program they call ACTIVE, Accelerating COVID-19 Therapeutic Interventions and Vaccines. So it's about drugs and it's about vaccines. And they already have partnership with over 18 companies, with public institutions, with universities, and with philanthropic organizations. The ACTIVE programs aims at harmonization at all levels to ensure the comparability of vaccine candidates. So, harmonization. Harmonization in terms of having a master protocol for all of the clinical studies. Prioritization of vaccine candidates. So the most promising vaccine candidates are given priority. An adaptive design, as for the WHO, which means that you can add vaccine candidates as you go along to your study. A single placebo control group, we talked about that. Then common clinical endpoints. Those are the criteria to evaluate efficacy and safety of your vaccines. Then a common statistical analysis by the same group of independent statisticians, and a common data and safety monitoring board. This board evaluates the data of your studies, and common laboratories for the analysis of your samples, using the same tests to analyze these samples to make sure that the results are comparable. And finally, the ACTIVE program by National Institutes of Health offers a trial network in the US and also globally, all over the world. Then there is another organization that you might want to know about. It's the CEPI Foundation. CEPI stands for Coalition of Epidemic Preparedness Innovation. It is an international organization financed by the European Commission, by individual member states of the European Union, also by the Bill and Melinda Gates Foundation. Those are the three big ones. WHO, then the NIH with the ACTIVE program, and the CEPI. Now we talk about money. Money is very important, as you might have imagined already. And there are extraordinary financial resources made available right now. I did a quick calculation. It's a bit simple, but it gives you an idea of the order of magnitude. So if the Bill and Melinda Gates say they will support 7 vaccine candidates, the NIH support 12 vaccine candidates, and CEPI 9 vaccine candidates, you end up with 28 vaccine candidates having already their financing assured. 28 vaccine candidates, if you take, for example, 500,000, let's say $500 million or $1 demi-milliard of US dollars, 28 vaccine candidates, you would have 14 billions, milliards en français, of US dollars made available, not upfront, obviously, but as you go along in development. This doesn't even take into account additional financial resources that come out of China or of other Asian countries. It also spreads to manufacturing, because what you need if you want to speed up development of vaccines is you need early and accelerated scale-ups for manufacturing. And that entails important financial risk, because basically what it means is that you are only in phase 1 and you already start building up manufacturing capacity for test vaccines where you don't know what will ever be commercialized. So these are important financial risks, but that's the only way of proceeding rapidly towards the coronavirus vaccines. You just need to build up from very early on sufficient manufacturing capacity. And that is quite a challenge. But all these things are happening. And so I think we have good reasons to think that there might be a coronavirus vaccine available in good time. Now, to finish my conference, there's one last aspect. If there is not a coronavirus vaccine available in time, what will happen? I mean, the coronavirus disease has two phases. In the upper respiratory tract, the coronavirus causes respiratory symptoms that are relatively mild in about 80% of people, comparable to symptoms of a common cold. However, in the lower respiratory tract, the coronavirus can cause a respiratory distress syndrome with a high mortality, comparable to the mortality you would observe with SARS coronavirus. Overall, the mortality, or to be precise, the infection fatality rate has been estimated at 0.75% in a recent meta-analysis. You know, meta-analysis is an analysis that looks at published studies and then analyzes them to get an average information on the parameters of interest. Perhaps I should also explain what an infection fatality rate is. Generally, what you look at is you say, so many people have tested positive, and there are so many deaths, okay? But that does not give you an idea about mortality, because those that tested positive are not all the people who got infected with the virus. Those who have tested positive are just the tip of the iceberg. The numbers of those who have got infected obviously is much larger than those who have tested positive, because not all of them who got infected were tested. So this is a type of mortality, what we call infection fatality rate. I was talking when I said that the meta-analysis came to an average of 0.75% mortality. Now, our topic is, if there's no vaccine available, what is going to happen? The most likely scenario is that the virus will rapidly spread and infect a major part of the world's population within 1 to 2 years. After that, the virus probably will not go away, but will continue to circulate in a permanent fashion. Individuals who have developed an immunity against the virus may perhaps not be completely protected in case of reinfection, but then will probably only present mild symptoms compared to the symptoms of a common cold. There is one of these common cold coronaviruses, you might know there are 4 in particular that we know of. It's coronavirus OC43. And coronavirus OC43 perhaps could show the way, because coronavirus OC43 only causes mild symptoms, symptoms of a common cold. But certain researchers think that 130 years ago, in 1889, when the virus crossed the species barrier from cows to humans, it actually was much more virulent, of a virulence comparable to that of the novel coronavirus. Because in 1889, you have a pandemic with 1 million deaths. And those researchers think that this pandemic then was caused by coronavirus OC43. Now, can we be sure that the novel coronavirus will follow the path taken by coronavirus OC43? What is the degree of protection and the duration of immunity acquired in the course of infection with the novel coronavirus? We don't know for the time being. However, even if a coronavirus vaccine is not available in time, it will nevertheless be advisable to continue the development of certain vaccine candidates to obtain safety and efficacy data, and to be able to constitute a stock in case of future need. Clearly, humanity has an urgent need for vaccine technologies that can be rapidly adapted to any infectious agents. So my presentation ends here, and now I'm very excited and looking forward to listen to your questions and hopefully to find a good answer to them. Thank you for listening.
So I hear large applause, so I'm very happy about that. Thank you so much, Gerard, for this very nice and exhaustive overview. I think many things have become clearer to most of us. And we open now the discussion. I turn my head towards Kim Mao just to check whether our discussants from China have joined.
Yes, I'm here. Hear me well? I'm here.
Yeah.
Yes, I'm here. So I'm in contacting with our Chinese animators. So unfortunately, they are still in meetings, but they sent me questions here, so I can transfer the questions to you. And also, I received a question from YouTube channel, so I will also forward to Gerard. So the first one.
Yeah. Could you please also just shortly remind who are our discussants from China, because maybe there are some persons in the audience who didn't attend the webinar when they were talking about epidemiology.
Sorry, Gilbert, I cannot hear you well. I cannot hear you well. So
no, I think Gilbert said something about epidemiology, no?
How is it now? Can you hear me?
Yes, now it's better,
yeah.
yes.
Yeah, so before you transfer the questions, could you please introduce once again our colleagues from China, because there might be some people who didn't attend the previous webinar where they were the guest speakers.
Yes, of course. I will explain to you the two animators that we have invited to join today's discussion part. One is Dr. Wang Weibin. So he is the chair and the professor in the Department of Epidemiology of Fudan University in China. And his research interest is majorly focused on the transmission of infectious disease, including COVID-19, and the impact of vaccination program. This is why we would like to have him with us and kick off the panel discussion part. And another animator we have invited is his colleague, the associate professor of epidemiology at Fudan University School of Public Health and Fudan Global Health Institute. So is Professor Lu Yihan. And so he is also work on the public health department and especially on the statistics. Yes. So this is two animators we have invited, and they are still busy, so they asked me to transfer their questions to you. If you allow me to put the questions on the full screen, so I will. So like this, our students and the audience can see it well. I will just, sorry. Okay. So this is first question from Professor Wang. So he would like to mention that in the literature or in the discussions with some researchers, he fear that people are reporting lower than expected level of viral antibodies in people who have recovered from COVID-19. So this possibly implies that any immunity gained will be just temporary. So he wondered if the vaccine of COVID-19 will be able to stimulate a stronger response and give a longer lasting immunity. So what is your opinion? Yes.
Right. I think it is important not to jump to conclusions all too fast. I think the first part of the question is some researchers are reporting lower than expected levels of viral antibodies in people. That is a fact. And first of all, we need to understand that fact. Actually, there is, I think it's a Chinese study where they have studied 149 people who got infected with COVID-19. And actually, in 1/3 of those, there were undetectable antibody levels. So they could not detect COVID antibodies in these people, although they had confirmed COVID disease. So what can you make out of this? I mean, first of all, you could say, okay, it might depend on the test, because if the test is not very sensitive, you can have false negatives. I mean, not detect antibodies while there are antibodies. That is one possible explanation. Another possible explanation is that perhaps antibodies are not the whole story. Remember that I told you that the type of immunity you would want to achieve is where you have both antibodies and certain immune cells. You need immune cells to clear infections, to kill infected cells, and to stop viral replication. This is largely antibody independent. Then there's something else to be considered. That's also what people are discussing, because the human immune system has a very old ancestral part. We call it the innate immune system. That is an immune system that is immediately ready to react, and that does not work with antibodies. Then we have what is called adaptive immunity. What you want to stimulate with the vaccine is adaptive immunity. Antibody production is part of adaptive immunity. So that adaptive immunity has developed much later in evolution. And in that adaptive immunity, they would find certain white blood cells, such as T lymphocytes, and you would find antibodies. Now, antibodies. I mean, this is not the first instance where people who have been vaccinated or even got infected show low antibody levels. I mean, antibodies are produced by cells. And cells may have memory for producing antibodies, even if there are very little circulating antibodies around. I mean, we know that after vaccination, after 10 or 20 years, people might have undetectable antibodies, but they might still have a certain degree of protection because they still have memory cells that are capable of producing antibodies once their immune system gets stimulated. So this, you cannot immediately jump to the conclusion, as is suggested here by the question, that implies or possibly implies that there is a temporary immunity. I mean, this is premature. However, what we know from SARS is that those who survived the SARS coronavirus, you know that it appeared first in 2002, 2003 in China, they have long-lasting antibodies, antibodies that last for many years at very good levels. MERS coronavirus is a bit less certain. People keep antibodies. I mean, what you would expect is that over time, antibodies decline, which does not mean that they may not raise very rapidly if the cells that produce them are stimulated. So it is interesting, however, to see that here you have people who had a recent infection with coronavirus, and even after a recent infection, you don't find very high levels of antibodies, because in the same study I mentioned, 1/3 of people had undetectable antibodies, and most of the other ones had relatively low titers of antibodies present in their blood. And that after, let's say, a relatively recent episode of of COVID infection. So this could imply that antibodies are perhaps not the whole thing, and that we need not worry too much about antibodies, always knowing that the tests may perhaps not have been optimal to detect the antibodies. Will the vaccine be able to stimulate a stronger response? I mean, with the vaccine, what we want as a response, this is a problem. For example, with the messenger RNA vaccine, where now Moderna, they published the data on 8 healthy volunteers. I mean, I don't know what kind of conclusion one ever could draw out of the data of 8 healthy volunteers, but immediately their stock market price rose to unexpected highs after this. Yes, so they have, for example, animal data. No, and you have data in humans, in 8 healthy volunteers. And there they are very happy to announce that you find the same amount, levels of antibodies with their Moderna messenger RNA vaccines as you find in people who naturally have recovered from the virus. But now we know that people who recover from the virus tend to have very low levels. So to be able to reproduce very low levels with your vaccine doesn't actually tell you whether your vaccine is effective. So I think there's a lot to be said about antibodies. As I said, antibodies are produced by cells, and they may not produce high levels, but they might produce high levels upon challenge with the virus. So not having high levels does not necessarily mean there is no immunity. But antibody also cannot, perhaps are not the whole answer. You need antibodies because they protect you from the beginning. Either from getting sick or from developing severe disease. But as I also mentioned, once you have COVID infection, antibodies are not enough. You need cellular immunity. And there's a very complex interplay between antibodies and cellular immunity, always talking about this more evolutionary, more recent adaptive immune system. If you look at the innate immune system, who knows? Perhaps in some people, the innate immune system is largely sufficient to control coronavirus infection, which could actually also be good news.
Yes, I think that we can fortunately keep these questions still for next week, because just by the way, I want to mention that this Thursday, we have opportunity to invite a virologist from Wuhan. So his research interests are innate immunity and viral infections. And he has emphasis on studying the interactions between host cell innate immunity and virus infections. So this will be an interesting follow-up discussion. I will enjoy the moment to just ask this simple question again.
Oh, you should do that, absolutely, because I think to think about antibodies is very simplified. So you have an interplay between antibodies and between T cells within the adaptive immune system, but then you have the innate immune system, and you have an interplay. You have lots of connections between the innate immune system and the adaptive immune system. So it is an oversimplification to rely on antibodies. They are easy to measure, and when you cannot measure them, people start getting quite nervous. But they certainly are not the whole thing.
Yeah. So now the second question. The second question, so it's also from Professor Wang. So he wondered, is distributed production ready to help, and what other measures can be taken to ensure that a vaccine reach all those in need?
Yes, what I mentioned is that actually, even with remdesivir, I won't comment on remdesivir today, but already the company producing remdesivir, the Gilead, they are producing large amounts for free, actually, also for countries where populations may not be able to afford them. I think there are philanthropic organizations who very much are committed to see that those vaccines are made available. But there are other possibilities. I mean, if you look at India, India traditionally has just copied drugs, irrespective of intellectual property. Now, that can be justified because there's more than 1 billion people living in India, and most of them would simply not be able to afford drugs unless there were drugs that are relatively inexpensive, because without intellectual property tied to them, I mean generics basically, and produced on a large scale. So if you have a drug developer, let's say in Cambridge, Massachusetts, in the US, the drug developer can very well grant an authorization to manufacture the drugs to certain manufacturers in India or in other countries, just to make sure that they will have sufficient vaccine or drug, I mean, here we are talking about vaccines, available for their populations. They can do that even without any claims on cost. I mean, they cannot do that for the whole world. I mean, they wouldn't have costs themselves, because if India produces the vaccines on their platforms, and if Cambridge, Massachusetts, the company, doesn't ask them for any money, just gives them the instructions how it can be manufactured, then there is no direct loss in terms of financial resources committed to manufacturing in India by the company in Cambridge, Massachusetts. There's only an indirect financial loss if you would imagine that there is this market in India where perhaps you could sell your drugs to Western prices. But I think with the WHO, with international organizations, with the philanthropic organizations, also with NIH, they are all willing to see that there is a prioritization as to who will get access first to vaccines, and that obviously resource-poor countries are part on the list and on top of the list. These are promises, and these are obviously initiatives. How it will all end up in the end, we will be able to observe. But I think people are very much aware of that problem. I also think that, imagine, like the Oxford vaccine, they really want, I mean, they're optimistic and said it might be able to produce millions of them already before the end of the year, if everything goes fine and safety and efficacy has been shown and sufficient participants in studies recruited. But even with millions of doses, you don't get very far. I mean, you need billions, you need thousands of billions. And that's also one of the reasons I said that there's probably a need to have a variety of vaccine technologies in development and later on available. Because with a single vaccine, you cannot achieve this.
Okay. I think that Professor Wang is trying to join us.
That's good.
Yes. Can you hear us, Professor Wang? I think it's not easy today. Professor Wang, we don't hear you. It's not easy today, so I will put the question, the next question from Professor Lu. I will, please allow me first before the configuration of the system is optimized. The question from Professor Lu is, this new coronavirus vaccine research and development has become a global task force. So a lot of countries participate in and spend a lot of money. However, we also know that the design, development, and production, as well as testing of each vaccine in the past have been a very long cycle. So as far as I know, normally we need 5 years to make a vaccine, the fastest one. But now, in order to fight against COVID-19, we have pressed it under 18 months, even less. So everyone wants to do a good job. But in case of problems, could it encourage anti-vaccine movement and something similar to vaccine hesitancy? What do you think, Professor Jihar?
Well, I think that actually there are several aspects that are covered by the questions by Professor Liu. As I showed, Professor Liu is absolutely correct that vaccine development generally has taken a lot of time, and the fastest vaccine ever developed was the one against Ebola virus, and that was in 5 years' time. Now, I also have given you very good reasons to think that specifically for certain vaccine technologies, there is realistic hope that we will be much faster this time. I won't repeat what I said during my presentation. However, so that's in terms of time. However, when you talk about vaccine hesitancy, you could also say that everything is a double-sided sword. So if you are very fast in developing your vaccine, some people might not trust it. They might just think that you might have taken shortcuts, that perhaps, you have had less participants in your studies, or that you have prematurely granted emergency approval, or that there has been fast-track review without really looking at all the data. I mean, you always have this kind of conspiracy theories and also of worries, actually, that the right choices are being taken, because it's always a risk-benefit calculation. On one hand, you want to be fast. On the other hand, you should not take undue risks. I can tell you that the FDA, for example, doesn't accept final study reports, because for every study you do in the vaccine development, companies submit a final study report with all their data, conclusions, and results. Now, the FDA doesn't accept that. The FDA wants the raw data of all your studies within development, and they analyze themselves, the experts, these raw data. They do their own statistical analysis. So normally these things are done in a very responsible way. But yes, people who are prone to vaccine hesitancy might actually think that the fast development might actually scare them. So whatever you do, you can never do it right. Now, you were talking about 18 months or 2 years. The question is, with respect to vaccines, obviously now with lockdown measures and with sanitary measures taken, the spread of the virus has slowed down, but the question is for how long can we continue these measures, particularly if we think that the pandemic will continue to spread. We don't know. But the anti-vaccine movement, I mean, there's quite a good analysis of why people hesitate with respect to vaccines. One of them is to say, is it really necessary to have my child vaccinated? Because many vaccines are administered in childhood. Is it not better to have a natural infection? Is the immunity that my child will acquire with natural infection not a better one than the immunity achieved with the vaccine? Then there are those who believe in conspiracy theories that, the virus was invented by drug companies, and now the governments with the drug companies just want to develop their drugs and their vaccines. You can never do very much about these conspiracy theories, but I think what is certainly needed is to be extremely transparent to the general public as to the way in which these trials have been done, run, and analyzed.
Yes. So in the past, as far as we know, that we still not have one previous vaccine existed for preventing coronavirus infection. So we are looking forward to it, but we are not sure. I remember in the last seminar I discussed with Laetitia this question, and with Professor Wang and Liu. So in New England Journal publication recently, they mentioned that a research report from Harvard Medical School they reported that maybe the vaccine will not finally be effective as we expected. And this disease will be unfortunately stay with us for the next 5 years. So we think we need to be aware of this fact and just do the risk management of all measures. Yes. So let me show you the next question, and this is the last one. If Professor Wang cannot be back, yes, he has some problem of the audio, so he just try again. So this is the last question from our YouTube channel. Christina Meyer asked, how can we guarantee that a future vaccine will be affordable and available worldwide? For example, low price barriers, as with many other drugs.
Yes, I mean, actually, I already answered that question, but I can come back to it for a moment. I think what is happening now is that there is an international coordination, and drug manufacturers are actually willing to participate in these international partnerships, and not to do it on their own, because there are many advantages for them. They get the money, they get access to manufacturing capacity, they get access to knowledge. These international programs are very much aware of the importance that vaccines be available on a worldwide scale. As I already mentioned, it is very likely that we need a variety of vaccines according to using different vaccine technologies to meet a global demand for a vaccine. When it comes to developing countries, what I mentioned just before was to say that there is a willingness that drug developers in our Western countries will actually give authorizations to manufacturers in developing countries to manufacture the drug. So then developing countries on their own platforms, in their own countries, can manufacture the drug or the vaccine in this case. It becomes then sort of a generic vaccine from the beginning because there's no intellectual property tied to it. And I think that is part of the deal. And that's also what large philanthropic organizations such as the Bill and Melinda Gates Foundation expect from these partnerships.
Okay, thank you. So I just received another question, a new question. Let me check if I can send to you. Yes, so from YouTube channel, student Maria Anon asked, are there any tests existing available to check innate immune system and for adaptive immunity, or is there research show some results?
No, I think there are biomarkers that allow you to look at the respective activity of adaptive immunity, certainly. As I already said, for adaptive immunity, what is very simple is to measure antibody levels in blood. What is already more complex for adaptive immunity is to measure activity of certain highly specialized immune cells. For the innate immune cells, you have other cells. You have phagocytes. I mean, phagocytes are cells that just engulf viruses to get rid of them. So the idea is to engulf them, they eat them, so they can digest them, degrade them, and that's the end of the virus. So you can obviously also look at these cells, I mean, every physician, when there's an infection, would look for white blood cells, because in infection, white blood cells go up, and the most frequent white blood cells are neutrophils. They're called neutrophils. They generally go up if there's an infection, and they are also phagocytes. So these are things you can very well look at, and I think they will be looked at. I also think that this is the topic of next Thursday's presentation, as I understood you.
One part. So,
okay.
because next Thursday, yes, at the end of the webinar, I will inform everyone the program. So I just want to inform you that yes, I just received another question from the same student. So let me know. Yes, because we have a very active discussion on the YouTube as well. So. Okay, so she wondered what are the negative effects for people of the adjuvant with aluminium in vaccine. Yeah, this is also a discussion for the part B, if I remember. Yeah, so please.
Yes, well, basically, I mean, if you aluminium is a very traditional, aluminium salts, you know, aluminium salts are very traditional adjuvants, and they are not necessarily the adjuvants that will be used, certainly not with the novel vaccine technologies. For example, I told you that for messenger RNA vaccines, you would rather use liposomes. I mean, liposomes are little spheres out of fat, and within these spheres, you would pack your vaccine, or you have lipid nanoparticles. I think that nowadays most adjuvants that will be, I don't actually think aluminium adjuvants will be used. I mean, it's difficult to say, but I don't think they rank very high among adjuvants that are nowadays available. I mean, previously with aluminium adjuvants, obviously, if you receive a vaccine dose, the dose of aluminium is quite low. I mean, there are many sources of aluminium in our food, in our environment, where you're exposed to aluminium. And if you compare that, the exposure of aluminium you have in a vaccine, then certainly your exposure to aluminium in the vaccine doesn't rank very high compared to your overall exposure to aluminium. Also, there are many drugs that contain much more important levels of aluminium, drugs that you take every day. In contrast to a vaccine where you get perhaps one shot or a maximum 3 shots of the vaccine.
Yes, I totally agree with you on this point, because in the past, in France or also in other countries, a lot of people debate on this point, and a lot of people, they don't agree that the small kids, the baby receive a certain vaccination because of this adjuvant aluminium. But I discussed with the pediatricians, so they told me that the baby can die from the disease, but they rarely die from the vaccine. Indeed. So yeah, so the dose is always the key point. Yeah, so I'm happy that you point out this key point here. This is also applied for other diseases.
That is a good question, because it is a general question. I mean, it comes back to what we said, is that you want to be totally transparent as to the way, particularly this accelerated way in which vaccines are developed right now. There are novel vaccine technologies. I mean, novel technologies, anything new seems frightening to many people, because they are not used to it. It's new, they don't understand it. We were talking about viral vector vaccines. Perhaps this is something very frightening if you look at it from hearing it from the first time. But if you take a non-pathogenic viruses, what is the risk? I mean, we are constantly exposed to viruses. All our lives through, we have been exposed to viruses. And every single virus will get into our cells and get our cells to produce viral proteins. It is what just happens all the time. So if now we have got the information into our cell to produce a viral protein, that is just what happens all the time. Except that in this case, it is intentional, because we want this to happen. But if you look at it in a more global way, we are just imitating what nature does all the time.
Yes, thank you. So we go to the next question. Unfortunately, our audience want to ask still a lot of questions, so let me show you the next one. So Ladani, she wants to know if the trial phases of a study for a new vaccine are accelerated. How big is the risk?
Just give me a second. I must answer a phone call.
Okay. So yes. So Gilbert, you want to have some comments on today's webinar?
Well, I'm fascinated that we get back and back again to this debate whether we should proceed with vaccinations or not. And as it was said by both Gerard and yourself, when there is, in general, the question to treat or not to treat in medicine, we should always think over what is more dangerous, disease or the vaccine. And talking about the COVID disease, I expect that some months ahead we will have an answer to the question and know that the disease is more dangerous than the vaccine. And if we look back, there is no more any child getting measles, which regularly killed young children. There is no young pregnant lady who doesn't get measles any longer. I always mix it up. The measles is the one that kills, and the other one, rubiola, is the one which provokes congenital malformations or even fetal death. So these are diseases that don't exist any longer in our areas owing to vaccination. We shouldn't forget that WHO was able to eradicate smallpox owing to massive worldwide vaccinations, that we don't see any longer any poliomyelitis in Europe thanks vaccination. So I think the debate is quite easy. Gerard is back to answer the question.
Yes, I think, thank you, Natalie, for asking this question. So I must make sure that I correctly understand the question. So the question is, the trial phase of a study for new vaccine are accelerated. How big is the risk of slowing down the development of other vaccines in case this strategy goes wrong? I'm not 100% sure that I get the question. I mean, what we have right now is that we have this international coordination for vaccine developments that brings together companies that develop vaccines, but also the public institutions, universities, and other parties. And they have a policy of prioritization. So at any time, the most promising vaccine gets most support. So you could have a very promising vaccine at the beginning, and then the data don't look good for that vaccine. In this case, this particular vaccine will just lose rank and go down in priority. And meanwhile, another vaccine that has much more promising data in the studies will be given priority. I'm not sure whether that's the question. The development of other vaccines, or are you talking about developing vaccines for other diseases rather than coronavirus disease? That actually could be some concern because, I mean, there's a certain manufacturing capacity and also, you know, in terms of conducting studies, in terms of public and private funding available, and coronavirus disease is now considered top priority. And so a lot of these capacities go to the development of coronavirus vaccines, and development of vaccines for other diseases certainly are given less priority right now. That certainly is correct. And then there's an aspect in case the strategy goes wrong, safety problems. Well, if you have safety problems, you will probably stop your study because you have a regular review of your data by an independent data review board. and they follow up on your data. And if your data don't look good or seem to entail a risk for participants in a study, then very simply, the study can be stopped. Nobody is interested in having safety problems or in developing a vaccine with safety problems, because if we do that, that will destroy any kind of trust in vaccine developers by the general public. I mean, vaccines are based on trust. You must trust that they are safe. You must trust that they are effective. You must trust that sufficient data on efficacy and safety have been generated before they have been commercialized. I think that to jeopardize this kind of trust would be a total disaster, because that would be the end of people's willingness to get vaccinated, or at least of many people's willingness to get vaccinated.
Okay, so we don't have any more questions, except if Gilbert has one question.
Well, actually, no questions, but many thanks. Thanks, first of all, to our guest speaker today, who made a tremendous work to bring all this knowledge together in a very comprehensive review. Thank you for those who were attending and who participated, in particular with asking questions. And of course, a big hello to our friends from China. I'm sure that it is not the last time we have met today. And well, thank you to you, Xianqing, for being always present and managing this. What we should say, perhaps publicly, is that Xianqing joined our team on 1st of April, when the university was already closed. So she has worked like mad for our institution without having been inside so far. So somewhere we are looking forward to have a welcome party for her arrival sometime.
Oh, great, yeah.
When we can
I'm looking forward to it.
And then we never see him on the screen, but all this is possible due to the technical skills of our friend Philippe, who was actually the initiator of this click meeting technology and who has followed us over these 2 months' time. So the final word would be to say, don't miss the following event where we will discuss the issue of maternal to fetal transmission of the virus by a top expert from Wuhan University, Professor Hu Wei. So enjoy the evening. We are deconfining. Don't forget that the virus is still around. Wear your mask, wash your hands, and remain in good health, please.
Yes, just as Gilbert mentioned, I put the practical information of Lex webinar of this week in Thursday here on the screen, so everyone from the YouTube channel can also have access to the information. So the title of the webinar is How to Obtain Evidence of Vertical Transmission in Viral Infected Pregnant Women, Reflection on COVID-19 Epidemic. So this is a very interesting topic that we cannot ignore, that the life goes on and people give birth. So if in case that the woman who will give birth infected by COVID-19, how the things will be managed. So the time is 2 to 4 o'clock in European time, and 8 to 10 o'clock in the evening for people live in Asia countries. And is this Thursday, 28 May. So the speaker is Professor Hu Wei from the Basic Medical College. So he is the Vice Dean of University of Wuhan in China. So we are very lucky to have two experts in this field in Luxembourg as well with us. So animators will be Dr. Stephanie Kreis, Assistant Professor of University of Luxembourg, and Dr. Jean-Claude Schmidt of our Medical Director from Ministry of Health. So thank you again for your participation of today's webinar, and stay safe. See you soon.
See you soon. Thank you.
See you soon.
On est entre nous maintenant?
Not yet.
Not yet.
Philippe.